we're going to talk about probably the most studied mechanisms of all mechanisms in organic chemistry, and that's the Fischer esterification of a carboxylic acid with an alcohol. Now, I'm going to try and keep things rather simple, but you do need to have a carboxylic acid to make this happen. And you will have an alcohol in solution. That's usually your solvent. And we also have an acid, and the acid that you use does need to be an oxy acid. That is important. Now, as you look at these different pieces and parts, they've studied this extensively, and one of the ways in which they've studied this is through isotope labeling. They know which oxygen ends up where, and we're going to talk about a variety of different options here. But you think about the fact that you are dealing with the strong acids. Acids are proton donors. You're going to have a proton available. And if you think about where the proton can go, proton is going to go to an electronegative atom. There are 3 oxygens that it could potentially go to. And we're going to look at each of these 3 different options. The correct choice will be to give it to the carbonyl oxygen. We'll talk about that. Another option would have been to give it to the carboxylic acid's OH oxygen, or to have given it to the alcohol. We want to look at all of these options here for a minute. We use different colors so that we have different pathways to follow. Now the red one is the correct one, but I do want to show you each of these pathways and then we'll continue on. Now let's look at the green pathway first and why that is not a good option. If you had protonated the alcohol, you have made a weaker acid-base pair. The problem is you don't have anywhere else to go at this point in time with something like this. You don't have any nucleophile to add to your methyl group, your CH3 group, to kick out water. There isn't anything. And so you kind of get yourself at a dead end. It isn't that this oxygen can't be protonated. It can be. But if it does happen, you're going to have to go right back where you came from. This is non-productive chemistry. So instead of doing this, maybe let's talk about the blue route. This also does not occur, but let's talk about it. So if you want to think about the blue route, which is also wrong, by the way. This is a mistake I often see students make. They protonate here. Now, I'm not saying that oxygen couldn't be protonated. It could be. And some of you go, Sister Stoll, that is a fantastic leaving group. We could kick out water there. This could be good. I want to do this. And some of you will say, I want to do that. Now, what's the problem with that? Well, it's the bond you're breaking. If that oxygen does add to the carbonyl carbon, pi bonds are weaker than sigma bonds. You're not kicking out your leaving group. You break pi bonds before you kick out sigma bonds. So this isn't going to work for you. In fact, it's kind of crazy. It doesn't make sense because you do have a good leaving group over there, but you have a pi bond. And so you've got 2 problems. Which route do you choose? Do you want to go, do you want to go and kick out the pi bond, or do you want to kick out the better leaving group? It's question mark. You can't do this. So this is not a good idea either. Don't go this route. If, on the other hand, you follow the red route, which is the correct route, you have something really awesome happen. And we're going to show you what happens. So these other oxygens technically could be protonated. So we're going the red route now. If you'd protonated the carbonyl oxygen, you will make this intermediate. This is really great. I'm going to draw in the other species that are present here for a moment. Now, I hope some of you are hopefully thinking about why this is really great. The reason it's really great is because it has resonance. Now, you can break this pi bond and give the electrons to the oxygen. As you do that, you can get a second resonance structure. And this isn't all you can do. The other oxygen can act as an electron pair donor toward the carbonyl carbon, and you can get yet a third resonance structure. There is an awful lot of charge delocalization of the positive charge here. The other two options, the blue option and the green option, there is no resonance delocalization of the positive charge. It has nowhere else to go. Nowhere. But with the red option, you have 3 resonance structures. So this is a much more stable cation, and this is where you want to put the proton due to the fact that there is resonance delocalization of the positive charge. This is much better. Now I'm going to redraw this in black on the next slide, but this is why this is the particular cation you want. There's resonance stabilization of the positive charge. And you can literally work with any of these 3 resonance structures you want, but I'm just going to draw the first one again. Okay, now you do have this HSO4 minus. It is not going to do anything else the rest of the time. And you have an alcohol. Now, things that are positive are attracted to things that are negative. I hope you know that by now in chemistry. And the alcohol has a negatively charged, well, it's not negative, but it's a partial negative oxygen in terms of the way the bonds are oriented. That oxygen can act as a nucleophile because this complex is now activated. It's got a positive character. And if you remember, back here you have a resonance structure that has a full positive charge on that carbonyl carbon. This carbon very much would like something to bind to it. And as that happens, the pi bond breaks. You can use any one of those 3 resonance structures you wanted. If you use the one I just underlined, just add it directly. The other 2 will do exactly what I'm showing here. But it will react. And as it does so, you're going to make a tetrahedral intermediate. This one's a protonated tetrahedral intermediate, but it is a tetrahedral intermediate. So this is the protonated tetrahedral intermediate that we have. What's going to occur now is we're going to do a proton transfer through solvent from the oxygen that has the positive charge to one of the other 2 oxygens. Because right now, this is the best leaving group. And we actually want this group to stay. We want one of the OH groups to go away. All of these oxygens are about the same chemically. They're all about the same basicity. And so it doesn't matter which one has it. So proton transfer through solvent is what's going to happen now. And our solvent in this particular case is the alcohol. So we have more alcohol present. And this reaction really is in equilibrium because you basically play ping pong with a proton. It doesn't matter which of these oxygens has the proton as long as it's on one of them. But this is the proton transfer to the solvent. Oftentimes on an arrow I will write PTTS. This means proton transfer through solvent. And this is the proton transfer to the solvent. So we are transferring the proton to the solvent and we will make the neutral tetrahedral intermediate. Now we are going to want to reform a pi bond to the carbonyl carbon and kick out a leaving group. But right now you don't have a good leaving group. You'd be kicking out a hydroxide, a methoxide, or a methide. They're all really strong bases if you make a double bond right now. And you can't do that. You're an acid. So you've got to protonate one of these oxygens. And it can literally be any one of these three. If you pick the OCH3, you're going right back where you came from. So don't pick that. It's non-productive chemistry. But it happens. Just don't do it. Pick one of the other oxygens. But one of them will act as an electron pair donor and pick up the proton. As that happens, the electrons are given back to the oxygen. So this is the proton transfer from solvent. And you get another protonated tetrahedral intermediate. Now we have a good leaving group, and it can be either the oxygen with the hydrogen or the oxygen with the methyl group that acts as the electron pair donor to make the pi bond. It doesn't matter which one. I just think it makes more sense for it to be the OH oxygen, but it can be the other one. As it forms a pi bond, you can't have 5 bonds to carbon, so you kick out your leaving group. This is what you have, and what's really special about this particular cation is, again, it has resonance delocalization of the positive charge. So I'm going to try and draw those for you. That pi bond can break, and as it breaks, oxygen is the more electronegative atom, so carbon would end up with the positive charge. And the other oxygen can also act as an electron pair donor to that carbonyl carbon. And you could get this resonance structure, which is why I said back here that it doesn't matter if it's the OH oxygen or the OCH3 oxygen that makes the double bond. It doesn't matter. It just makes more sense, I think, to see it from the OH oxygen. But there are 3 resonance structures here. So you've got a lot of charge stabilization. Basically, the reason this is at equilibrium is you don't make a weaker acid-base pair at all during the course of the reaction. Not at all. But you make an equivalent acid-base pair. So going on, you would now need to neutralize your acid. Typically, to do this, you add a little bit of base. But we're going to use the solvent to do this process for our purposes right now. I'm just drawing one of those 3 resonance structures. And then you have your base come in. I'm going to use an alcohol. It's probably actually not a strong enough base to do this favorably. Usually you do add a little bit of weak base solution, like a sodium hydroxide solution, weak concentration to do the job. But we're going to show it with this. So you pick off your proton, give electrons back, and you end up with your ester. Now, if you look really carefully at some of these other slides, notice that the products that are formed are also going to include water. That's very important. So if you want esterification to happen, you want to drive a carboxylic acid and an alcohol toward the ester and water. The way you would do this is by making sure that the acid you're using is concentrated to begin with and you have an excess of the alcohol. If, on the other hand, you want to drive the reaction toward the carboxylic acid and the alcohol, you would do this reaction in dilute water. Not dilute water, dilute acid in water. You'd want to make sure that there's a lot of water present, and if you do that, you'll do the same reaction in reverse, and I'll show that to you a little later. But it's very important that you understand this operates under Le Chatelier's principle. And since water is a product formed during the course of the reaction, you'd want to dry that water out. Usually you use molecular sieves, and we have a lab that we do with this, and I don't think we're drying it out since we're not trying to make this perfect, but if you were really concerned about this, you would dry out the water to make sure that you drive the reaction to completion and produce take the carboxylic acid to the ester.